Hello everyone, here is Dr. Shuting Xu from GenScript. Today I would like to introduce you a novel high-throughput test for detection of SARS-Cov-2 neutralizing antibody. So before the talk, I want to give you a brief look of how today's webinar will be. First, I will give you some simple facts about SARS-CoV-2, and then I will introduce you about our cPass kit. Afterwards, of course, I will explain to you why cPass kit is important to use and why you should choose it. In the end, I will explain to you the importance of neutralizing antibodies in the whole combat with COVID-19. And of course, I will end with a short summary for today's talk. So about SARS-CoV-2, I think till today there will be no one says, I don't know about this virus. According to WHO, until yesterday's data, there are currently 130 million cases reported that is infected with COVID-19. More than 2.8 million deaths associated with COVID-19 were also reported globally. Unfortunately, while we're talking, this number is still rising rapidly. So how does SARS-CoV reach this level of pandemic? First of all, as we can see from the scheme of the virus on the left side, SARS-CoV-2 virus, also known as COVID-19, it has a very simple viral structure. It encodes S protein, E protein, covers with envelope. On the top of the envelope has M protein, and also within the virus, it has viral RNA and associated with M protein. So how the virus enter our host cells? It has to give the credit to the virus S protein, also known as spike protein. Within the spike protein, there is a part called receptor binding domain, also known as RBD domain. This domain can interact with the host cell surface receptor ACE2, and with this interaction, allows the virus into the host cells and thus replicate in the host cells. If the interaction is blocked with a neutralizing antibody, this virus cannot invade the cells anymore. Until today, so how do we detect the SARS-CoV-2? As we already know, there are different ways to detect the virus infection and at different time frames. The most common way, as we see, is the PCR test. This allows you to determine the viral load in the infected patients. Besides the PCR test, we also have the so-called serology test, also known as antigen test. It can be either a quick test or it can also be an ELISA test. Both tests allow you to detect post-infection of the antigen response within the patient. So what is cPass? What can cPass do? cPass Neutralization Antibody Detection Kit, also known as SVNT, detects the circulating neutralizing antibodies against SARS-CoV-2 that block the interaction between the receptor binding domain, as we know RBD domain, of the S protein with the ACE2 receptor. So, as we already know what's the definition of cPass ELISA kit, how does it really work? The cPass kit is designed for detection the neutralizing antibodies with the patient plasma or serum samples. So once you have your samples ready, you can just take 10 microliter of the samples and inoculate with the kit provided RBD domain reagent. This RBD domain is conjugated with a reporter protein HRP. So as you can see from the figure, the neutralizing antibodies, when it existing in the samples, it will block the RBD domain trimer, which interacts where the green color indicated non-neutralizing antibodies either does not bind to the RBD domain at all, or it could also possibly bind to the other parts of the RBD domain. After the inoculation time, we transfer all the samples into our 96-well ELISA plate. Those plates are coated with the ACE2 receptors on the well bottom. We allow them to fully interact with each other at 37 degrees. With this temperature, you will allow the unblocked RBD domain to bind to the ACE2 receptor steadily. As you can know, afterwards, we can wash all the RBD domain away, which was blocked or neutralized by the neutralizing antibodies. And then we can read the plates with the signal of OD and the readout. So you can see this mechanism of cPass kit is quite easy. So why should I choose cPass? What's so special about this kit? As you know, before cPass kit, there are other ways to determine the neutralizing antibody within the samples. One of the most conventional way or traditional way is so-called virus neutralization assay. This assay needed the live virus attendance, and because of that reason, it takes 2 to 3 days to perform the assay, and also it needs the S3 lab because we need to use the live coronavirus. Additionally, to perform this assay, you need a highly trained expertise to do the experiment and also to interpret the data. This assay could be quite expensive due to the production of the virus and performing of the lengthy experiment. With the cPass kit compared to this traditional assay, it is much faster. It just takes up to 2 hours for one performance and can be highly automatable. Also, this work doesn't need S3 lab because it can be performing in any laboratory settings, as it just needs an incubator and an ELISA reader. It can be done, of course, in a high throughput way. As you know, it's quite easily performed. And also, this assay is much more consistent. As you see, it is only with the ELISA kit. So this is the most conventional way, but what is the gold standard of the neutralizing assay? As we know, the plaque reduction neutralization test. As you see from our manual data, we compared our kit results with the most gold standard up PRNT50 and PRNT90. Both assays showed 100% agreement with the cPass kit. Therefore, the specificity and sensitivity of our kit compared to this plaque assay is perfect. So at this moment, I guess you also have the question, what's the difference of cPass kit and other serology tests that determine the quantity of IgG or IgM? So as you can see here, with our assay, we involved the receptor coded on the bottom of our plate. Due to this reason, that's also why we call the surrogate virus neutralization test. We can really detect the functional neutralizing antibodies against the COVID-2. And you have the readout with the RBD domain binding to ACE2. When the neutralizing antibody works, there will be no signal at all. Serology tests that claim they could interpret the neutralization functions also, they have typically the RBD domain coated on the ELISA plate. With that, you can see from the scheme that maybe they are detecting the neutralizing antibodies, but it could also give you false signal when the antibodies binds to the other parts of RBD domain instead of exactly the trimer that interacts with a receptor. In order to show this in a more statistic way, we also performed an internal test. As you see here, the blue bar shows accumulation of S1 protein as we use antigen test, and the red line indicating the neutralizing activity using our cPass kit, so they are not really correlated. Additionally, with traditional antigen tests nowadays, they are mostly focused on IgG and IgM. However, with this assay, they could simply missing an importance of IgA. And cPass kit is detecting the total neutralizing antibodies against the S protein of SARS-CoV-2. It not distinguish IgG, IgM, or IgA. So we are also detecting IgA in our test. Studies already shown that IgA dominates the early neutralizing antibody response to SARS-CoV-2. This figure showed you that post the infection of COVID-19, within up to 2 weeks, the IgA dominates in the antigen response in this blue line. Another figure from this same research showed you that the purified IgA has a much higher neutralizing ability compared to the IgG, which conventionally people like to detect post the infection of SARS-CoV-2. Importantly and interestingly, here today we have some feedback from our current customers told us that cPass kit could also be potentially used for detection of saliva. As we all know that IgG is mainly existing in the saliva samples, so a saliva test in cPass could be an alternative for the future to determine the neutralizing antibody level in the patients. So besides all that, what else that cPass can offer to you? Of course, as I mentioned before, cPass is a very fast tool and easy to handle tool to detecting the neutralizing antibodies. Additionally, cPass is also species independent. The characterization from the animal model and to human clinic studies can be performed with cPass kit. Till today, there are many studies already use the cPass kit for animal samples, including mouse, non-human primates, rabbit, bat, dog, cats, and pangolin, and so on. And very important of the cPass kit is this is virus specific. There is 100% no cross reaction with especially influenza A and B. As we all know that influenza has quite similar symptoms as COVID-19. So to distinguish them with a simple antigen test is also important and crucial for the kit, and cPass can offer you that. Importantly, our cPass kit already received many important regulatory approvals. For instance, we have the HSA mark in Singapore. We received the CE-IVD mark in Europe, and we also have the FDA EUA authorization. Of note, cPass here today is still the only one FDA EUA authorized commercial ELISA kit for detecting neutralizing antibodies against the SARS-CoV-2. Besides the current kit, we have extended our potential of cPass. We launched recently a new neutralizing antibody standard that allows the kit for quantitative assay, so you can have your result in unit per mil instead of percentage of inhibition. So the antibody standard is already on process of obtaining the FDA EUA as well as CE-IVD mark. So let's come to the last topic. Why is it important to detect the neutralizing antibodies as well compared to the other, detection of antigen? Some studies already shown that there is a poor correlation between antigen test and neutralization. As you see from this data offered by this particular study, they use different kits to detecting the samples for IgG test, and then they also use a conventional neutralizing assay. And you see the correlation between the neutralizing activity and the IgG test is really bad. Thus, the paper or the research group concludes that the commercial serological assays have poor MPA for SARS-CoV-2 neutralization, making them imperfect for neutralization test. Additionally, another independent research group showed that the positive binding ELISA may not mean positive neutralization result. As we already showed you before, but this study showed that within 218 participants that have already positive ELISA result for the binding, IgG binding assay, only under 60% of the patients have positive for neutralization assay. Another importance of why we have to study neutralizing antibody is due to the individual immune response. This study that published quite recently gave us a first hint of how and why we have to detect the neutralizing antibodies post-infection and validation. So the study is performed in Singapore, and they have accumulated 517 samples. And this cohort study lasted up to 9 months long. So they are detecting the neutralizing antibody response post the natural infection. And you can see here, we can easily distinguish the response within 5 groups. The first group is so-called negative groups. They have almost no neutralizing antibody response post the infection for the whole 9 months time. The second group, which the research group called them the rapid warning, they seem to have a high enhancement of the neutralizing antibody immediately when they exposed to the virus infection. However, this neutralizing antibody response seems not lasting within the virus infection and rapidly dropped to almost negative up to 2 months or up to 2 to 3 months. The third group, the green group, is so-called slow warning. So these start with very high immune response after the infection. However, this neutralizing antibody response can be last quite long, and still 150 days they're still above the positive line. And then the fourth group, which is also a major group called the persistent group, they are from beginning till the end obtained a very high amount of neutralizing antibody response. And then the last group, which is quite special and quite individual, is they have relatively medium level of response in the beginning and somehow have a higher response later. It could due to other second infection or other diseases, which they need to find out later. So this study already gives us a first hint that, even though everyone can be infected with the virus, but that doesn't mean that you are immune post-infection, because it could belongs to any of these 5 groups that you don't know. And some people could have a longer term of protection and some have a very short term. And so with this study, the research group also proposed a way to identify the neutralizing antibody levels. So they draw these 3 lines. Under the 30% of inhibition, they call it negative, also suggested by our kit. And then they have under 50% of inhibition, they call it a low immune response. Between 50% and 80% of inhibition, they suggest it is a medium level of neutralizing antibody response, and above 80%, they say it is a high immune response, and most likely they are protected. For your information, the cPass kit is also have an official application already. It is acknowledged by the FDA that cPass can be used for qualifying the convalescent plasma quality. as well as the kit can detect the samples have above 68% inhibition, and this convalescent plasma can be used for treatment. So in the very end, I would like to give you a unpublished, very preliminary data from our collaborations that they have performed post-vaccination evaluation and monitoring those patients after the vaccination and how they respond to the vaccine. So as you can see, this group of study, those patients have been vaccinated with either Moderna or Pfizer vaccine, and they have quite different response, especially after the dose 1, as you see from the left figure. So after the dose 1, some people may think, oh, I'm already vaccinated, I'm safe. As you see, this is not the case. Some people already have high inhibition after the vaccination, but many people are varies post the first vaccination. And after the second dose of vaccination even, there are still quite a few individuals have medium level of response, either Pfizer or Moderna vaccine. And importantly, this study also gives us a glimpse of how different ages respond to the vaccination. As you can see from the right-hand side figure, after the first dose, the younger group seems to have a much better response to the vaccine, while the older elderly group have much slower response. And see, post the first vaccine, many of them doesn't show any positive cross the positive threshold. And even after the dose 2, and some old people have less response. And however, you can also see young patients could also have low response post the second vaccine. So here comes my summary of today's talk. As you already see, the presence of binding antibody may not guarantee the presence of neutralizing antibody, and the neutralizing antibodies are important biomarker for possible immunity, and cPass, also our serology our test, are the best choice for determining the neutralizing antibodies in the patient samples. However, with the cPass kit, there are still quite open questions waiting for us to answer. Is everybody vaccinated means everyone has immunity? Is there cured from SARS-CoV-2 means that you are safe? This is a long way to go for the science globally to answer those questions. Well, cPass can be the tool for you to help you to answer the questions. With cPass, you can use it for validate your vaccine and drug development. As you see that the cPass is designed for plasma and serum samples detection for neutralizing antibodies, but from the mechanism, you can understand this can be also a perfect tool to validating the antibody drugs that blocking the viral entry. It can be also a perfect tool for that, for instance. Secondly, how we define the herd immunity. This is still a debate in many, many countries nowadays. I think it's still a global problem because we could not just understand yet that what is the threshold for reach the herd immunity. Well, cPass is a tool to help you to define this threshold line. And we hope soon with the global effect, we can understand this threshold better and we can draw the line much faster and allow the global traveling back again. And the third, of course, cPass can help you and help your country to monitoring the vaccine efficiency. As I already showed you in our last unpublished data, that even though everyone can be vaccinated, but doesn't mean everyone is protected very well with a different level of immune response. So with the cPass tool that you can better monitoring past vaccination, how the whole population is respond to the vaccine. So if you have more questions, you can come to us to understand it better. So in the end, I want to thank you for your attention. And I mentioned before, if you have more questions and want to understand our kit better, you can come to us for more information. And thank you.